Hello friends, hope all of you are doing great. In this video, we are going to discuss about the blood-brain barrier and the circumventricular organs. If you have not watched our previous video about the cerebrospinal fluid, I strongly recommend that you watch that video before you proceed any further. So let's dive right in. In our last class, we have discussed about the cerebrospinal fluid. Today, we will first learn about the blood-brain barrier and then proceed to discuss about the circumventricular organs. The blood-brain barrier is the barrier between cerebral capillary blood and the cerebrospinal fluid. The concept of such a barrier was first proposed by the German Nobel laureate Paul Elrick. He did an experiment and injected tryptophan blue, which is an azo dye, in the bloodstream of a mouse. To his amazement, the dye readily infiltrated all the tissues except the brain and the spinal cord. But he incorrectly concluded that the CNS must have a very low affinity to the dye and hence it is unable to penetrate the cerebrospinal fluid and the brain. This is Paul Elrick in an old German currency called Mark. Nowadays, euro is used in Germany. By the way, he won the Nobel Prize for Physiology or Medicine in 1908 along with Dr. Mechnikov, who was also a great Russian scientist. They won the Nobel in recognition of their work on immunity. But back to our experiment about the blood-brain barrier. Around 2 decades later, after the original experiment, a student of Paul Elrick, Dr. Edwin Goldman, rejected the hypothesis of Elrick. Dr. Goldman repeated Elrick's initial experiment and got the same result. He then performed a second set of experiments where he injected the dye directly into the brain and CSF compartments. He found that the brain and the CSF readily takes up the dye. Hence, he concluded that the outcome has nothing to do with the affinity of the dye. He further proposed that there must be a barrier of some sort between blood on one side and the CNS on the other. But the actual anatomical locus was not discovered until the development of the electron microscope. Finally, it was found that the location of blood-brain barrier is at the level of the CNS capillaries. This is just a representational diagram. You can see the CNS capillary wall on the left side and the capillary wall of the mucosa of the intestinal tract on the right. In the case of the CNS endothelium, you can notice the green structures in between the endothelium, which are the tight junctions, and they are absent in the case of the other capillary. The most important thing which is present in the CNS vasculature and which act as a blood-brain barrier is the tight junction. We will discuss about the tight junctions in a moment. Now focus on the capillary on the right side. Roman 1 represents the paracellular route through which larger polar molecules can pass between the interendothelial gaps to the intestinal fluid and vice versa. Roman 2 represents the transcellular route through which exchange can occur via 2A, which is transcytotic vesicular transport, and through 2B, which are fenestrations or windows. Now look at the next diagram. you can identify the out of proportion CNS capillary. Here in the CNS capillaries, the paracellular route is effectively blocked by the tight junctions, and the transcellular route is also blocked due to the absence of vesicular transport and also due to the non-availability of the fenestrations. There are other mechanisms through which transports are blocked, but we will not go into further details just now. Among all the blood-brain barriers present in the CNS vasculature, the most important is the tight junctions, represented by the green structures in between the endothelium. The tight junctions are made up of proteins such as occludin, which is the first identified membrane protein within the tight junctions. Next, we have claudins, which is a family of 24 proteins. Further, we have junctional adhesion molecules or JAMs, which belong to the immunoglobulin superfamily. The capillaries are also covered by a vest of end fit of astrocytes. Together with the other mechanisms discussed earlier, they produce the blood-brain barrier. Ultimately speaking, crossing of the brain capillaries is possible through the oily membrane of the endothelial cells. Thus, lipid-soluble materials, for example, carbon dioxide, oxygen, lipid-soluble drugs, can cross the blood-brain barrier. The permeability across the blood-brain barrier follows two major mechanisms. The first is the carrier-mediated transport, as you can see here, and the other is the receptor-mediated transcytosis. Functions of blood-brain barrier. It maintains a constancy of environment in and around the brain. It prevents escape of neurotransmitters. It protects the neuron from harmful substances that may be present in the blood. Circumventricular organs. Certain areas of the brain have fenestrated capillaries and due to their permeability are said to be outside the blood-brain barrier. These areas are located mostly around the third ventricle and hence are called circumventricular organs. The brain contains 6 circumventricular organs. CVOs comprise specialized brain regions where the blood-brain barrier is interrupted to allow cerebral neurons to interact with the circulation directly. Some CVOs are sensory while the others are secretory. Sensory CVOs, represented by the green color, include the subfornical organ and the organ vasculosum of the lamina terminalis, both of which are associated with the hypothalamus. The area postrema is a brainstem CVO. The second categories of CVO are secretory or neurosecretory CVOs, represented by the red, which includes the median eminence, which is a part of the hypothalamus, the neurohypophysis or the posterior pituitary gland, and the pineal gland. The neurosecretory organs secreting polypeptide neurotransmitters are allowed to enter the circulation freely, while the others are chemoreceptor areas for sensing the blood-borne substances that must be allowed to reach the brain. Thank you for your attention. If you have any doubts, suggestions or questions, please write in the comments section below. I'll be happy to help you. See you guys in the next video.